Hi guys, so we'll start the session. I'll share my screen and you can let me know if the screen is visible and if my voice is audible. Wait a second. Sorry. Okay. Okay, so can one of you unmute and let me know if my screen is visible and if I'm audible?
Yes, sir. Yes, ma'am.
Okay, okay. Thank you. Thank you. Okay. Okay, so we'll start. So, good evening, everyone. Today's week 7 of these live sessions. So, in these live sessions, what we do is that we basically talk about some assignment problems from this course, Genetic Engineering Theory and Application, taken by course instructor Professor Vishal Trivedi of IIT Guwahati. So, in these sessions, basically, what we do is we discuss some core concepts behind those questions and we try to figure out the answers, and we discuss it in detail so that it's easier for us to solve similar type of other questions as well, right? So today's the 7th live session for that, and we try to keep these sessions as interactive as possible. So if you have any question, you can raise your hand either um you can raise your hand either on the Google Meet, or you can unmute and let me know, and you can type in the chat box as well, and if I know the answer I'll reply right now. If I don't, then I'll try to read more about it and get back to you by the next class, right? So we'll start with the first question for today. One second. Okay, so in glycolysis, two ATP molecules are utilized by the enzymes dash and dash. So what are the two enzymes? The options are A. Hexokinase pyruvate kinase, B. enolase pyruvate kinase, C. Hexokinase phosphofructokinase, and D. Aldolase enolase. Which one do you think is the correct answer? I'll wait for 2 minutes. One of you can try and answer me.
Ma'am, option C,
Kinase
hexokinase
and
and
phosphofructokinase. Okay. Okay. So I'll wait for 1 more minute, then I'll move on. Okay, so the answer is right, hexokinase and phosphofructokinase. So we'll talk a little bit about the different steps of glycolysis and what is what before we move on to the next question. So we know that the normal blood glucose concentrations, normal plasma glucose concentrations roughly vary from 4 to 8 millimolar, right? And there are various glucose transporters that we know of. So there are various forms of glucose transporters. We have various, as you can see, the variants are GLUT1, GLUT2, GLUT3, and GLUT4. So the tissue locations of these are different. So what what do these glucose transporters do exactly? So that's what we'll talk about, and we'll talk about the importance of glycolysis, obviously. So glucose transporters are members of the solute carrier gene family, and they are membrane proteins which permit the facilitated diffusion of hexoses and polyols, like including inositol, glucosamine, ascorbate, into and out of the cells down their concentration gradient, right? So, and We know that glucose reaches the brain by two types of transporters: sodium independent glucose transporters and sodium dependent glucose transporters. Okay, and in the brain, the transporters GLUT1 and GLUT3 are known to play a major role in glucose uptake via facilitated diffusion. Okay, so there are basically two mechanisms for glucose transport across cell membranes. In the intestine and renal proximal tubule, glucose is transported against a concentration gradient by a secondary active transport system in which glucose is co-transported with sodium ions, right? So as I said, glucose transporters are a wide group of membrane proteins, and it facilitates the transport of glucose across the plasma membrane, and this process will be called as facilitated diffusion. Now, because glucose is a vital source of energy for all life, these transporters are actually present in all phylum. All phylum. So the GLUT or the solute carrier gene family are a protein family that is found in most mammalian cells, and 14 GLUTs are encoded by the human genome. Okay, and GLUT, this is a type of unipolar transporter protein, right? So we know that most non-autotrophic cells are unable to produce free glucose because they lack the expression of glucose 6 phosphatase, and thus are involved only in glucose uptake and catabolism. Now, usually produced only in hepatocytes. In fasting conditions, other tissues such as intestines, muscles, brains, and kidneys are able to produce glucose following activation of gluconeogenesis. And what happens in mammals, so these glutes are they contain 12 membrane spanning helices with both the amino and the carboxyl termini exposed on the cytoplasmic side of the plasma membrane, and these GLUT proteins transport glucose and related hexoses according to a model of alternate conformation, and which predicts if the transporter exposes a single substrate binding site towards either the outside or the inside of the cell, and binding of the glucose to one side can provoke a conformational change associated with transport and can release glucose to the other side of the membrane, right? So moving ahead, these are the glucose transporters. So this is basically nothing but a Michaelis Menten curve. If you can see, on the x-axis we have the substrate concentration, on the y-axis we have a Vmax. Okay, so these are the Michaelis Menten curves for the different uh, GLUT transporters. Oh, and this particular range is the normal physiological range of glucose concentration. Now, GLUT4, as we know, is recruited to cell membrane by insulin dependent translocation. So the increase in number of GLUT4 transporters in the cell membrane increases the speed of glucose transport. Insulin stimulates glucose uptake in fat tissue and skeletal muscle, right? Now, talking about glycolysis or the uh, Emden-Merhoff Parnas glycolytic pathway. So this is the stoichiometry. We have discussed a little bit about glycolysis in the last class. So glycolysis is used to provide energy in the form of ATP, obviously, and intermediates for other metabolic pathways, and glycolysis occurs in cytoplasm. Now, when there's aerobic conditions available, like oxygen is available, oxygen is required to reoxidize NADH to NAD plus via the ETC, electron transport chain in the mitochondria, right? But when there is anaerobic conditions available, NADH is reoxidized to NAD plus in a reaction in which pyruvate is reduced to lactate in humans, lactic acid fermentation, or ethanol, alcoholic fermentation in case of yeast and other microorganisms. Now, glycolysis. So, if you see, glycolysis gets converted to pyruvate via glucose gets converted to pyruvate via glycolysis, and the pyruvate under aerobic conditions can undergo the Krebs cycle or the TCA cycle, tricarboxylic acid cycle, or the citric acid cycle, whatever you call it, and then it can undergo oxidative phosphorylation of the mitochondrial membrane and then ultimately give rise to CO2 and H2O. But under anaerobic conditions, the process followed is fermentation, either lactic acid fermentation or alcoholic fermentation. Okay. These are the various structures that are, you will see the relevance in you'll see the relevance of these structures in um, glycolysis. So these structures actually copied from somewhere. Okay. Now, glucose plus ATP will get converted to glucose 6 phosphate plus ADP, and the enzyme will be glucokinase or hexokinase. Okay, it's an irreversible, highly exergonic, and regulated step in glycolysis. Right. So glucokinase is basically what it does is it's a glucose sensor. So if you see, this is also Michaelis. No, this is also Michaelis-Menten curve, where on the y-axis we have V, on the x-axis we have substrate concentration. Okay. Now glucose stimulated insulin secretion in pancreatic beta cells. You see the glucose is getting inside cells by a GLUT2 transporter. It gets converted to glucose 6 phosphate and then ultimately to pyruvate, right? And for pyruvate, the glycolysis happens in cytoplasm. Then from there, if there is aerobic condition available, it will go inside the mitochondria, and finally Krebs cycle or oxidative phosphorylation will happen, and all of that. Now this will, there will be insulin secretion because of the glucose present, right? So that is why we call it glucose stimulated insulin secretion in pancreatic beta cells. Okay, so the body recognizes that there is glucose in the system and we should secrete insulin. So glucose metabolism determines secretion of insulin. So GLUT2 is a high km, okay, high efficiency glucotransporter. So it is not rate limiting, correct? So that is what we know about GLUT. GLUT2 is one of the most popular, like known glucose transporters, and that is what we know about like in Michaelis-Menten equation. If you know, so Km, which is also known as the Michaelis constant, so the substrate concentration at which the reaction rate is 50% of the Vmax, right? So Km is basically a measure of the affinity an enzyme has for its substrate. As the lower the value of Km, the more efficient the enzyme is at carrying out its function at a lower substrate concentration, right? So it indicates basically that substrate concentration attains half its maximum velocity when enzyme catalyzes the chemical reaction. Now glucokinase has a high Km and is rate limiting. Reaction velocity is roughly proportional to glucose concentration. Therefore, glucose metabolism and insulin secretion in beta cells is roughly proportional to glucose concentration. Now then the glucose 6 phosphate will get converted to fructose 6 phosphate, right? So that is done by phosphoglucose isomerase. From fructose 6 phosphate with the help of ATP, it will get converted to fructose 1,6 bisphosphate plus ADP, or phosphofructokinase. So this is an irreversible reaction, highly exergonic, regulated step in glycolysis, and this is one of the main regulatory enzymes of glycolytic pathway. Okay, so there's 2 ATP that is spent, actually not made, spent in phosphorylating glucose and fructose 6 phosphate. Then we have the enzyme aldolase that catalyzes the reaction from that helps in the reaction fructose 1,6 bisphosphate to dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, right? Then we have triose phosphate isomerase, which basically helps interchange amongst these two forms. Either it will be a DHAP or glycerol G3P. So either it's dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So triose phosphate isomerase, basically what it does is it helps maintain a balance between the two because it helps in interconverting these two forms. Now next step will be conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate, and this is carried out by the enzyme glyceraldehyde 3-phosphate dehydrogenase. Okay, and the product form is basically a mixed anhydride of phosphoric acid and a carboxylic acid. So energy from oxidation of glyceraldehyde 3-phosphate to the carboxylic acid is captured in the formation of the acyl phosphate linkage, right? Next reaction is basically 1,3-phosphoglycerate plus ADP, which gets converted to 3-phosphoglycerate plus ATP, and this is carried out by the enzyme phosphoglycerate kinase. So this is known as substrate level. This is known as substrate level phosphorylation. So if you check the ATP balance over here, 2 ATP is spent in phosphorylating glucose and fructose 6 phosphate, and 2 into 1 ATP generated in the phosphoglycerate kinase step. Now, what do you mean by substrate level phosphorylation? So, it's basically a metabolic reaction that generates ADP or GTP by transferring a phosphate group from a high energy compound to ADP or GDP, right? And it's an exergonic reaction that can occur in cytoplasm or mitochondria and under both aerobic and anaerobic conditions. Okay, substrate phosphorylation basically means the direct formation of ATP or GTP by transferring a phosphate group from a high energy compound to ADP or GTP, right? Whereas oxidative level phosphorylation means is a cellular process that harnesses the reduction of oxygen to generate high energy phosphate bonds in the form of ATP, adenosine triphosphate, right? Moving on. Then 3-phosphoglycerate gets converted to 2-phosphoglycerate using phosphoglycerate, like the enzyme is phosphoglycerate mutase. After that, 2-phosphoglycerate gets converted to phosphoenol pyruvate and water with the help of enolase. And then phosphoenol pyruvate will, with the help of ADP and the enzyme pyruvate kinase, will get converted to pyruvate and ATP. This is another substrate level phosphorylation step. So again, irreversible, highly exergonic, and it's a regulated step in glycolysis. So now if we talk about the ATP balance, so 2 ATP that was spent in phosphorylating glucose and fructose 6 phosphate. So 2 into 1 ATP generated in the phosphoglycerate kinase step and 2 into 1 ATP generated in the pyruvate kinase step. So 2 molecules of ATP are generated from 1 molecule of glucose in glycolysis. So this is how the stoichiometry looks like. 1 glucose plus 2 ADP plus 2 PI, that is inorganic phosphate, plus 2 NAD plus. It will give rise to 2 pyruvate, 2 ATP, 2 H2O and 2 NADH and 2 H plus. Now the 3 reversible enzyme catalyzed reactions are the hexokinase step or the glucokinase step, the phosphofructokinase step and the pyruvate kinase step. Now, as I said, in yeast and many other organisms, pyruvate with the help of hydrogen ions will get converted to acetaldehyde and CO2 under the action of pyruvate decarboxylase. And obviously, acetaldehyde and NADH plus H plus can get converted to ethanol and NAD plus under the action of alcohol dehydrogenase. One second. Okay. In mammals, sorry. In mammals, the pyruvate along with NADH plus H plus will get converted to lactate and lactate dehydrogenase under the action of lactate. So it's seen that under anaerobic conditions, that is in rapidly contracting muscles. So if you have seen that if you're exercising a lot or running for a while, you'll see that your legs will start paining, right? So that's because of the accumulation of lactate in your calf muscles. Okay, so that's what happened. That's what happens when there's lactate accumulation in your body. So this is schematic to show the fates of pyruvate generated by glycolysis, either lactate via lactic acid fermentation or acetyl CoA, oxaloacetate. Basically, both will go into the TC cycle, alanine and all of that. Okay. Now, the regulation of these metabolic pathways. So, the flow of intermediates through metabolic pathways is controlled by mostly four mechanisms. Okay. Number one is substrate availability. Number two is allosteric activation or inhibition. Number three is covalent modification of enzymes. And number four is induction or repression of enzyme synthesis. Right. So, if we talk about the signals, the energy state, ATP is high energy signal, ADP and AMP are low energy signal. Okay. So, availability of biosynthetic intermediates, citrate and alanine. A metabolic state of the whole organism. Insulin, signal of the fed state. High blood glucose, signal of the fed state. Glucagon is signal of the fasted state, right? And epinephrine is signal of stress. So metabolic regulation of these biochemical pathways makes the physiological sense. So control of flow through a pathway is often distributed among several enzymatic steps in the pathway. Like if you see in reversal hepatocyte in the fed state, what is happening? So there is insulin being secreted, right? There is glucose. Glucose is being transported inside using GLUT2 transporter. Insulin is there. Insulin receptor catches insulin, and which basically induces the intake of glucose via the GLUT2 transporter, right? Which ultimately gets converted to glucose 6-phosphate and either to glycogen, or it undergoes glycolysis to form pyruvate and then might be fight again form lipids. So this is hepatic glucose uptake and utilization. In case of fasted state, starved state, when you are not eating, what happens is that the glucagon is released, which is basically recognized by its receptor. Then it comes inside. What it does is that it basically in general induces the breakdown of glycogen in the body, right? So this process known as gluconeogenesis, because glucose is being prepared. So glycogen is converted to glucose 6-phosphate, which undergoes another reaction to form glucose, and this glucose is given out. So that is the hepatic glucose production. In case of skeletal muscle, what happens? In rapidly contracting state, the glucose will be taken in by GLUT4 transporter, right? In low ATP and high AMP conditions, and which will get converted to glucose 6-phosphate. Glycogen is also there, and it will ultimately undergo glycolysis and give rise to ATP. In resting state, however, GLUT4 is again taken inside and converted to glucose 6-phosphate, and then it gets converted to glycogen, and then otherwise it gets catalyzed via the glycolytic pathway. So as I said, this was the first step almost. Glucose plus ATP will be glucose 6-phosphate plus ADP, and the reaction is catalyzed by glucokinase and hexokinase. So glucose 6-phosphate is low phosphofructokinase. Glucose 6-phosphate is a negative feedback basically. So what it does is that it shows low phosphofructokinase activity. Okay, but in case of glucokinase, high blood glucose that is released, and high Km and insulin stimulation that will ultimately stimulate gene expression will give basically a rise. So this is only seen in liver. So glucokinase can be found to the glucokinase regulatory protein, and when this glucokinase is bound to the GKRP, it is sequestered in the nucleus and is inactive. So glucose promotes dissociation of GK from the GKRP, allowing GK to be transported to the cytoplasm where it is active in glycolysis. Next step is fructose 6-phosphate plus ATP to fructose 1,6-bisphosphate plus ADP, and it is done catalyzed by the enzyme phosphofructokinase, right? So the main regulatory steps of the glycolysis are like ATP, that's a high energy state, will act as a negative feedback. Citrate, abundance of biosynthetic building blocks, and H plus building of lactate will also However, AMP, which is a low energy state, and fructose 2,6 bisphosphate, which is a fat state, high insulin or glucagon ratio. So that will act as a positive feedback. Now, ATP is a substrate for phosphofructokinase, and ATP can also bind to an allosteric site. So in this case, from the conversion of phosphoenol pyruvate plus ADP to pyruvate plus ADP using pyruvate kinase, ADP is a high energy state in alanine, which is abundance of alanine, which is synthesized from pyruvate and is important in gluconeogenic substrate is and glucagon. This is the fastest state. They might act as negative inhibitors, whereas fructose 1,6 bisphosphate feed forward mechanism and glucose stimulating gene transcription will act in a positive way. Now, glucagon binding to the glucagon receptor, activation of the adenylyl cyclase, increase in CAMP, activation of protein kinase A, phosphorylation and activation of pyruvate kinase. So it occurs in liver in the fastest state. So there is every step that is dependent on its previous step. So we have talked about the major objectives over here. We are talking about glycolysis, a major oxidative pathway of glucose. We have talked about the main reactions of glycolysis pathway. We have talked about the rate limiting enzymes regulation, ATP production, aerobic and anaerobic, what happens, lactate or alcoholic, or it undergoes TCA cycle, whatever has happened. Pyruvate kinase deficiency can lead to hemolytic anemia. So all of these, most of these we have discussed. Now moving on, as I already told you in a summary form, if we can check this. So glycolysis is the main pathway for glucose oxidation. It occurs in cytosol of all cells. Now it is unique in that it can function either aerobically or anaerobically depending on the availability of oxygen and intact mitochondria, and it allows tissues to survive in the presence or absence of oxygen and skeletal muscle. RBCs which lack mitochondria are completely reliant on glucose as their metabolic fuel, and they metabolize it by anaerobic glycolysis. So if you see, this is the process of aerobic versus anaerobic glycolysis. Okay, aerobic glucose to glucose 6-phosphate to fructose 6-phosphate to fructose 1,6 bisphosphate to glyceraldehyde 3-phosphate dihydroxyacetone phosphate, then 1,3 bisphosphoglycerate, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenol pyruvate and pyruvate. Okay, in case of anaerobic glycolysis, it will also get converted to pyruvate, but then it will move ahead to lactate. Whereas in case of aerobic glycolysis, it will go forward for TCA cycle and oxidative phosphorylation in the mitochondria. We have already discussed this. Hexokinase is seen in basically most tissues, whereas glucokinase is popular in in hepatocytes. So this is the first step where D-glucose is converted to glucose 6-phosphate. Okay, by means of the enzyme hexokinase or glucokinase, and ATP is being converted to ADP, like energy is being spent. Next is fructose 6-phosphate, which basically converts to fructose 1,6 bisphosphate, right? With the help of ATP, which gets converted to ADP, and the enzyme is phosphofructokinase 1. Okay, so ATP and citrate will negatively inhibit this process, whereas AMP and fructose 2,6 bisphosphate will negatively enhance, the negative will act as a positive feedback for this process. So from fructose 1,6 bisphosphate, it can take two ways via aldolase, which is glyceraldehyde 3-phosphate or acetone phosphate, and they can interchange between each other using this transphosphate isomerase as well. Okay, we have talked about this slide. So glyceraldehyde 3-phosphate dehydrogenase. So for each NADH, 3 ATP will be produced by ETC, or the electron transport chain, in the mitochondria. That is, ATP are being produced. So then we come to the phosphoglycerate kinase, which is substrate level phosphorylation, and pyruvate kinase, which is second substrate level phosphorylation. So as I was saying, phosphorylation is the metabolic reaction of introducing a phosphate group into an organic molecule. So oxidative phosphorylation, basically formation of high energy phosphate bonds by phosphorylation of ADP to ATP, coupled to the transfer of electrons from reduced coenzymes to molecular oxygen via the electron transport chain. It occurs in the mitochondria. Whereas substrate level phosphorylation is the formation of high energy phosphate boundary by phosphorylation of ADP to ATP, uh, metabolic uh, or GDP to GDP, coupled to cleavage of a high energy metabolic intermediate like substrate, and it may occur in cytosol or in mitochondria. Right, so these are the basic differences in substrate level and auxiliary phosphorylation. So next we talk about pyruvate kinase. Okay, so what happens is that the pyruvate kinase is generally inactive, in inactive form. The PEP or the phosphoryl pyruvate will get converted to pyruvate only when pyruvate kinase is available. And for that, what they have shown in this schematic over here, this glucagon is being recognized by its receptor, which ultimately triggers adenylate cyclase to form CAMP, cyclic AMP, and PPI or inorganic phosphate from ATP, that will lead to an active protein kinase, and ultimately it will move ahead to the next step. So long time regulation of glycolysis. Giving insulin will be induction, giving glucagon will be repression. Okay, so if you can see in the first step also, glucose to glucose 6 phosphate, insulin is green plus, whereas glucagon is negative minus. In the next step also, increasing insulin will probably induce the process, whereas increasing glucagon will decrease. The middle steps are fine, then at the end step also, increasing insulin acts as a positive feedback, whereas increasing glucagon might not be that. So in summary, regulation of glycolysis, regulatory enzymes, irreversible reactions will be glucokinase, hexokinase, phosphofructokinase 1, pyruvate kinase. Okay, and regulatory mechanisms will be rapid short term, allosteric covalent modifications, so long term induction repression, and that is how these enzymes work. So if we talk about one of the enzymes and its deficiency, pyruvate kinase deficiency, hemolytic anemia. So what happens? There are altered enzymes, kinetics also decreases, enzyme stability altered, and response to activator also goes down. Okay, so what happens? The enzyme may show an abnormal Km or Vmax for substrates or coenzymes, and the enzyme may show the abnormal response to the activator fructose 1,6 phosphate. So enzyme activity or stability may be altered, uh, or the amount of the enzyme may be decreased. And we have talked about all of these aerobic glycolysis, how much ATP production happens. ATP consumption is 2, ATP production is probably net 8. Then aerobic glycolysis versus net ATP production. For energy investment phase, 2 ATP is taken, and energy generation phase, 2 ATP, and then 4 ATP and 2 NADPH are given out. NADH are given out. Then we talk about anaerobic glycolysis. So in this case, lactate dehydrogenase basically converts lactate to pyruvate, right? So the ATP consumed is 2 ATP, and the ATP produced is 4, and total is 4 ATP. Okay, so 4 minus 2 will be 2 ATP. Anaerobic glycolysis in RBCs, 2,3 BPG unit. We talk about that. Two three BPG shut. Sorry. So if we talk about this is 2,3 bisphosphoglycerate, which gets converted to only phosphate using phosphatase, right? So that is the role. Now glycolysis in RBC's production, we have already talked about this. Also ATP consumed and ATP spent and all of that. So this is summary. So take home message is glycolysis is the major oxidation pathway for glucose. It's employed by all tissues. It is the tightly regulated pathway. PFK1 or fructokinase 1 is a rate limiting regulatory enzyme, and glycolysis is mainly a catabolic pathway for ATP production, but it has also some anabolic features like amphibolic. And pyruvate kinase efficiency in RBCs results in hemolytic anemia. So this was a take-home message. The ATP gained from the process. So in case of catabolic reactions in general, complex molecules will give rise to small molecules. And in the case of anabolic reactions, simple molecules and energy will give rise to complex molecules. So as I said, the glucose is converted to two molecules of pyruvate and anaerobic reaction in cytoplasm. We have checked all this. We don't need to get into details. Overall, the first two steps of glycolysis, the phosphate to phosphate groups added, 6-membered glucose ring is isomerized into 5-membered glucose ring. Energy stored into ATP molecules is utilized to modify the structure of the glucose. We have talked about all these steps. The two glyceraldehyde triphosphate units are converted to two pyruvate units in phase 2 of glycolysis. So overall, the energy generating phase from 2 NADPH plus and 4 ATPs. So 2 ATPs are used in the phase 1 of the glycolysis and 4 ATPs are made in phase 2 of glycolysis. The net result is 2 ATPs, 4 minus 2. And the 2 ADDs formed are made in the cytoplasm and must be transported to the mitochondria to join the electron transport chain and make ATP. I talked about the fate of the pyruvate. Okay, so coming back to the question that we started with, in glycolysis, 2 ATP molecules are utilized by the enzymes dash and dash. So yes, the answer will be hexokinase and phosphofructokinase, right? So we'll wait for 3-4 minutes and then we'll move on to the next question for today. If you have any doubts regarding this question, you can ask, or we'll wait for 3-4 minutes and then only we'll move to the next question. Okay, so we'll move on to the next question for today. So moving on to the next question. Which of the following cell surface receptor is involved in the uptake of glucose? We have already talked about this. So it's glutes. So I kept this question because it was quite similar to the last one. I wanted to talk a little bit about the glucose transporters that we have because it might come in some competitive exams. So obviously it's glutes, right? So what is the importance of these transporters? What do they help in, and which process exactly they, matlab, which in which tissues or cell systems do these basically work in, and what are the different uh, isoforms of these glucose transporters that are involved? So there can be various types of transportation, right? So it can be either non-carrier mediated like diffusion osmosis, or carrier mediated like active transport and then uh, facilitated diffusion. So all that can be vesicle mediated like exocytosis, endocytosis, and endocytosis we can talk about pinocytosis and phagocytosis as well. Right. So So if we talk about non-carrier mediated movement, transport, so we'll talk about diffusion. Diffusion is the tendency for molecules to spread out evenly into the available space, right? So what happens? Substances diffuse down their concentration gradient. The difference in concentration of a substance from one area to another is basically what determines the diffusion. And the diffusion of a substance across a biological membrane is passive transport because it requires no energy from the cell to make it happen. Okay. Now the rate of diffusion can depend on either the magnitude of the concentration gradient or the permeability of the membrane, temperature. Like higher temperature is faster diffusion rate, surface area of the membrane can also be a determining factor, and microvilli, which increases the surface area. Now, the rate of diffusion of a gas into a liquid is directly proportional to the partial pressures of the gas above the liquid surface area of the available for gas exchange and the solubility coefficient of the gas, and inversely proportional to gram molecular weight of the molecules and thickness of the membrane. So the Fick's law basically is what. So Fick's law is of diffusion is a model that describes how particles flow due to diffusion. So it's used in many fields including biology, medicine and chemistry, and the law states that the rate of diffusion is proportional to the surface area and the concentration gradient like difference. So if you see this is the diffusion coefficient, this is the partial pressure gradient, this is the surface area, and it is basically this is inversely proportional to the thickness of barrier of the membrane, right? So what can be some of the examples where Fick's law can come into use? Like obviously drug absorption, right? Fick's law can explain how drug molecules will move from a higher concentration to a lower concentration until equilibrium is achieved. Or smoke propagation, and it can be used to model how exhaust from a smokestack mixes with the surrounding air, right? So that is, I think it was first proposition by Adolf Fick in 1855, and hence the name, on the basis of basic largely experimental results. So they can be used to solve for the diffusion coefficient, which is D over here. And Fick's first law can be used to derive a second law, which in turn is identical to the diffusion coefficient. Okay, so the first law, Fick's first law, states that movement of particles from high to low concentration, that is diffusive flux, is directly proportional to the particles' concentration gradient, which I already talked. And Fick's second law dictates prediction of change in concentration gradient with time due to diffusion. So diffusion process which will obey this fixed law is called a normal or Fickian diffusion. Otherwise, if it is not obeying it, it will be called anomalous diffusion or non-Fickian diffusion. Right? Again, then you move to osmosis. Now, osmosis is the diffusion of water across a selectively permeable membrane. Now, water diffuses across the membrane from the region of lower solute concentration to the region of higher solute concentration. So, if you see over here also, uh, how osmosis happens. So, in the middle, we have a selectively permeable membrane. So, water is over here, sugar is over here, right? So water is here we have a higher concentration of water but low concentration of sugar, where here it is exactly opposite. Now, water balance of cells without walls. Now, tonicity is the ability of the solution to cause a cell to gain or lose water. Now, water isotonic solution. Solute concentration is the same as that inside the cell. No net water movement across the plasma membrane. Or we can talk about hypertonic solution, where solute concentration is greater than that inside the cell and the cell loses water. And then you have hypotonic solution, where the solute concentration is less than that inside the cell and the cell gains water. So these are different conditions, which is isotonic, hypertonic, or hypotonic. So if we talk about live demonstration of osmosis, you can see over here, this is selectively permeable membrane, okay, and this is proper clean distilled water, and there is a 3% salt solution over here. This, if you keep it here, so the solution will keep, so there will be osmosis, the distilled water, since it has here the solute concentration is high, like salt concentration is high, so distilled water will start moving in through the selectively permeable membrane to here, and the salt solution will start rising, the level of the salt solution will start rising, and the solution will stop rising when the weight of the column will equal the osmotic pressure. Right? If we talk from physiological perspective, like physiological relevance, if you see this hypertonic solution, it's an animal cell. If the surrounding, if the inside is has a higher concentration of salt, H2O will come inside, like water will start coming inside, and it will ultimately get lysed. Cell will swell up and burst. On the contrary, if the pressure on both the sides are equal, it will maintain a proper isotonic system. Whereas if the pressure outside, so if the salt concentration is more outside, water from inside will start going outside and it will basically shrink or shrivel, exactly opposite of this condition. Now, in case of plant cells, it's basically same but little bit different because of the presence of a cell wall. So when there is salt concentration more inside, the water will start coming inside and it will become a turgid fat cell. If the pressure of water on both the sides are equal, it will be a flaccid normal cells. However, if the salt solution concentration is greater outside and rather than inside, water will go outside and it will ultimately give rise to a plasmolized solution. So if we want to talk about the difference between osmosis and diffusion, osmosis involves movement of solvent molecules, whereas diffusion involves also movement of solute molecules. Okay, so diffusion we talk about solute molecules. Like if you get inside a big room and you spray a perfume at one end, so the fragrance particles will diffuse through the membrane as through the room, right, and it will ultimately cover the entire room. So that is the movement of solute molecules, whereas in case of osmosis it is the movement of solvent molecules, right. In case of osmosis, the molecules move from lower to concentration of solute to higher concentration of solute, right. Whereas in case of diffusion, the molecules will move from higher concentration of solute to lower concentration, because in diffusion we are talking about the movement of solutes. So obviously they'll be going from a higher concentration to a lower concentration, right? If we talk with an analogy, if you have seen a local train, at peak office hours, the local train will be bursting with people, right? It will be so much crowded. But when there's an empty station, it approaches an empty station, it will ultimately, the people will get down. So that's what happens. People are moving from their higher concentration to their lower concentration. But in case of osmosis, we are talking about the diffusion of the movement of the solvent, right? So in this case, the molecules will move from their lower concentration to their higher concentration of solute. Sorry, molecules will move from the higher concentration of from lower concentration of solute to higher concentration of solute. That's the difference. So basically the concept is similar, but in both the cases we are talking about the movement of different items, right? In one case you're talking about the diffusion of solvent, as in the other case you're talking about the diffusion as a movement of solvent, and in the other case you're talking about the movement of solute. Osmosis needs semi-permeable membrane, as diffusion does not require any membrane. Osmosis example will be shaking of potato slice when kept in concentrated sucrose solution, right? Whereas diffusion, spreading of ink when a drop of it is put in a glass of water. Then we talk about reverse osmosis, a way to clean water out of dirty water or salt water by forcing water under pressure through a membrane. An example of reverse osmosis is the process of filtering polluted water under pressure. A plasma membrane is semi-permeable or selective membrane. So small hydrophobic molecules like O2, CO2, N2, benzene will move. Small uncharged polar molecules like water, ethanol, glycerol will also move across. Larger uncharged polar molecules like glucose, amino acids, nucleotides, these won't move across the membrane, just across the membrane. Ions also will not move just across the membrane. So for these, we need transporters or channels. So in case of facilitated diffusion, passive transport aided by proteins, ion channels that open or close in response to stimulus, like gated or non-gated channels. Then we have aquaporins for facilitated diffusion of water, glucose transporter, and bicarbonate chloride co-transporter. Now, if we talk about channels and transporters, they differ in stereospecificity, rate of diffusion, and saturation. So, if you see, in case of simple diffusion, it's basically a straight line. With increasing solute concentration, rate of transport will increase. In case of facilitated diffusion, if you see, it is following a like a Michaelis Menten type of curve, isn't it? So with increase in solute, it will jump. The rate of transport will jump high at the beginning, but after a point, it will reach a saturation or a plateau point. Like even after that, if you keep on increasing the concentration of solute, it will not increase the facilitated rate of transport as much. If you see the schematic, this is a hydrated solute that moves across the membrane. Okay, and this is a hydrated solute that moves across with the help of a transporter. Right. So if you see, in case of diffusion without transporter, the activation energy barrier is also very high. It's a simple diffusion, but if you have a transporter, the free energy barrier is not that high. So this is slide is to summarize the transport pathways that we are talking about for solutes across the cell membrane. So we either have simple diffusion, no requirement of any channels or transporters, etc., or we have facilitated diffusion, which requires a channel protein, or we have carrier proteins facilitated diffusion, or carrier proteins that will make use of energy. So that will be then termed active transport, because now they are making use of energy. Okay, so this is normal diffusion, higher concentration to lower concentration. This is passive transport, higher concentration to lower concentration via channel. This is also higher concentration to lower concentration via channel. In case of active transport, it is going from its lower concentration, if you see, the movement is happening of this square particles. If you see, it has a higher concentration on this side and a lower on this side, then why is it not moving from here to here? Because now it's moving the opposite way, but now we need to spend some energy because we are moving it across. For example, with an analogy, if the train or the metro is already if if as I said, the previous analogy only, if the train is very crowded and it comes and stops at a you know empty station, people will push you out and you will come out on your own. That is going from higher concentration to lower concentration. You don't need any energy. People will push you out of the crowded train. But if the train stops on a platform and you want to get inside the train, you're standing on the platform and you want to get inside the train, but there's a huge crowd in the train, then you need to push your way through. That's when you need to expend energy. Correct? To spend energy. So that's exactly what is happening over here. If you want to, this particle wants to get into the train which already has so many of similar particles, so now it has to spend energy for that. Okay. Now, if we talk a little bit about ion channels are nothing but membrane complex protein complexes. They provide a high conducting hydrophilic pathway across the hydrophobic interior of the membrane, and the channel or the post structure is said to catalyze the reaction of transporting charged molecules across. The catalytic site, central channel, is either open or closed. The open channel conformation can be compared to the transition state of the enzyme substrate complex, where ions are tightly associated to the catalytic site. Now, the conformational change between closed and open state is called gating. Ion channels can be classified according to which chemical or physical modulator controls their gating activity. Right? So it can be leakage ion channel or non-gated ion channel or gated ion channel. So the leakage ion channel is the non-gated ion channel. It's an integral membrane protein which is an ion channel within a cell's outer cell membrane. It is always open and permits the diffusion of one or more ions in the direction which is in accordance with their concentration and charge gradients. Then we have the gated ion channel. It's an integral membrane protein which is an ion channel within an excitable cell's outer cell membrane which opens and closes in response to some stimulus, that is membrane potential or voltage changes, the arrival and binding of a specific ligand or signal molecule like hormone, neurotransmitter and local hormone, or to mechanical pressure or to light energy. Now we have the voltage gated ion channel. So it opens and closes in response to stimulus, which is a change in membrane potential, like voltage, like propagation in excitable cells such as neurons, muscle cells, and gland cells. Then we have the chemically gated ion channel, which opens and closes in response to stimulus, which is the arrival and binding of a specific ligand or signal molecule like hormone, neurotransmitter, local hormone, etc. Then we have the mechanically gated ion channel, which opens and closes in response to a stimulus, which is the mechanical pressure or vibration. Sensory cell responding to touch, vibration, compression, or stretch. Or the light gated ion channel, which is a photosensitive excitable cell's outer cell membrane, which opens in response to a stimulus, which is the arrival of a photon of light energy. These the examples can be sensory cells, rods and cones responding to light in the retina of the eye. So this schematic shows this is the receptor of sodium. It's a chemical gated ion channel. It's closed. Now when the chemically gated ion channel opens, the neurotransmitter chemical attached to the receptor, sodium is pushed in, potassium is pushed out. And in case of voltage gated ion channel, is also closed. But if the membrane voltage changes, in this case the stimulus was chemical binds and it opens. In this case, if the membrane voltage changes, then that leads to the opening of the voltage gated ion channel, and it opens and sodium is pushed inside. Okay. Ionophore is basically a chemical species that reversibly binds ion it neutralizes the charge of ions and carries them down its concentration gradient. Okay, and it disrupts secondary transport processes and it kills microbial cells. So carrier ionophores that bind to a particular ion and shield its charge from the surrounding environment. This makes it easier for the ion to pass through the hydrophobic interior of the lipid membrane, like valinomycin potassium cation. Carrier ionophores may be proteins or other molecules. Channel formers that introduce a hydrophilic pore into the membrane allows ions to pass through without coming into contact with the membrane's hydrophobic interior. And an example of a channel former is gramicidin A. Correct? A channel forming ionophores are usually large proteins. Now, a little bit about the examples we have over here. Tetrodotoxin, tetrodotoxin, it is used by puffer fish and some types of newts for defense. It blocks sodium channels, and puffer fish is apparently very poisonous. Okay. Then we have saxitocin, which is produced by a dinoflagellate, also known as peptide, also very poisonous. It blocks voltage dependent sodium channels. Then conotoxin used by coal cells to hunt prey. Then lidocaine and novocaine belong to a class of local anesthetics which blocks sodium ion channels. Dendrotoxin is produced by mamba snakes and blocks the potassium channels. Iberiotoxin is produced by the Banthus stimulus. It is the East Indian scorpion and it blocks the potassium channels. And we have the heteropodatoxin, which is produced by Heteropoda venatoria, brown huntsman spider lyre, and it blocks potassium channels. So block acetylcholine receptors or prevents the opening of its ion channel. So like turbo or curate arrow poison in Amazon region, cobra toxin like of the cobra and the bungalow toxin of the crate. All of these are proper examples of these. Okay. Structure of a bacterial potassium channel was determined by X-ray crystallography once, and the channel is made from 4 identical transmembrane subunits which together form a central pore through the membrane. Now, negatively charged amino acids are concentrated at the cytosolic entrance to the pore and are thought to attract cations and repel anions, making the channel cation selective. Now, each subunit contributes 2 transmembrane helices which are tilted outward in the membrane, and together they form a cone with its wide end facing the outside of the cell where potassium ions exit the channel. Right? One second. Okay, now the polypeptide chain that connects the two transmembrane helices forms a short alpha helix, which is a pore helix, and a crucial loop that protrudes into the wide section of the cone to form the selectivity filter. Right? Now the selectivity loops from the 4 subunits and it form a short, rigid, narrow pore which is lined by the carbonyl oxygen atoms of their polypeptide backbones. Now because the selectivity loops of all known potassium channels uh, have similar amino acid sequences, it is likely that they form a closely similar structure. And the crystal structure shows that 2 potassium ions separated by about 8 angstrom. So mutual repulsion between the 2 ions is thought to help move them through the pore into the extracellular fluid. Okay. Now a little bit about the different transporters, carrier proteins that undergo subtle change in shape that translocate the solute binding site across the membrane. Uniport co-transporter, like it will transport different molecules, so it can be symporter or antiporter. Okay, so there are aquaporins, which are basically water channels. Different types of aquaporins is here that help transport different types of things. Transport proteins or carrier proteins speed the passive movement of molecules across the plasma membranes. There are glucose transporters. The GLUT1 is ubiquitous, it's present almost everywhere. The GLUT2 is prominent in liver, pancreas, islets of Langerhans, intestine, insulin release regulation, and all of that. GLUT3 is present in brain. Okay, GLUT4 is present in muscle and adipocytes. GLUT5 is intestine, testis, kidney, sperm, fructose. And GLUT6 can be spleen, leukocytes, brain, like no transporter function as such. Now, what is the chloride bicarbonate exchanger? Carbon dioxide produced by catabolism enters erythrocyte. Okay, if you can see over here. So from here it gets converted by carbonic anhydrous to here, and the bicarbonate dissolves in blood plasma. This is the chloride bicarbonate exchange protein is there. Okay, that transport protein, carrier protein that helps. Then in lungs, carbon dioxide leaves erythrocyte and is exhaled. Bicarbonate enters erythrocyte from blood plasma. So this is again in the first step, if you see, this is sodium potassium channel. In the first step, here sodium is low, potassium is high, right? So sodium is low over here, still sodium is being transported outside. So against the concentration gradient, so it requires energy, right? So sodium comes, 3 sodium come here, right? And then sodium is thrown outside in the extracellular fluid. At the same time, potassium goes inside, right? So potassium is also going against its concentration gradient. It is going from higher concentration, low concentration to high concentration, and potassium is thrown inside the cell. So both these require, it's happening together. It's antipolar because both are going in the opposite directions, and it requires energy. Now, a resting or non-signaling neuron has a voltage across its membrane called the resting membrane potential, or simply the resting potential, right? So the resting potential is determined by concentration gradients of ions across the membrane and by membrane permeability to each type of ion. Okay. We'll not go into very much details. So what are the examples of diffusion in living systems? For example, in lung, oxygen goes from airspace to RBCs. Adaptation happens in alveoli, moist lining, good blood supply. Then in lung, carbon dioxide goes from blood plasma to airspace. Alveoli, moist lining, good blood supply. Then digestive system goes from food molecules like glucose to food molecules will grow from stomach, intestine to blood. Uterus from mother's blood supply to fetus blood supply. Leaf from leaf cell to airspace. So what are everyday examples of diffusion? Smelling your mother's cooking from the kitchen while you're studying in the living room. As I give you an example of the fragrance that is sprayed at one part of the room, it will go to the other, across the entire room, it will spread across the room. Smelling garbage on the street before you can see the garbage truck. Smelling someone's perfume. Drop of ink spreading to color the water in a beaker without stirring. All of these. And in the digestive system, osmosis plays a key role in nutrient absorption. So osmosis allows your body to basically absorb these nutrients to the intestines and individual cells. Cells get rid of their waste by osmosis, and osmosis is largely important to plants because the root cells absorb water by this process. So what are the everyday day-to-day life examples of osmosis? When your hands are immersed in dishwater for a long time, your skin looks bloated. This is the effect of osmosis because now the water has gone inside. When you pour salt onto a slug and water diffuses and the slug shrinks as a result of osmosis. When you cook food and put sauce in the liquid part of your dish, some part of the solute moves inside the solid part of the food you are cooking. And the solid part could be anything. But the sauce is made up of solute and not water, so it will move into the food. All of these are examples of osmosis. Osmosis helps in the transfer of water and various nutrients between blood and fluid of cells. Plants also use osmosis to take in water and minerals essential for its growth. Reverse osmosis is a type of osmosis which is used to convert sewage water into clean drinking water. All of these. Will not go into very much details. Again, Fick's law we have discussed. So basically, diffusion refers to the random microscopic movement of water and other small molecules due to thermal collisions. And diffusion is also known as Brownian motion, named in honor of the Scottish botanist Robert Brown, who first observed spontaneous vibration of pollen particles under the microscope. And in the 19th century, German physicist Adolf Fick modeled diffusion as the movement of particles from a region of higher concentration to a region of lower concentration. And Fick showed that flux of particles was directly proportional to the concentration gradient, related by the factor D, the diffusion coefficient. So, because D reflects the flux of particles through a surface during a certain period of time, it has units of area per time. Then it was not until 1905 that Albert Einstein developed a comprehensive mathematical theory to explain Brownian motion and incorporate Fick's law of diffusion. So Einstein also incorporated into his random walk theory a principle of fluidic friction developed in the 19th century by George Stokes, and this became known as the Stokes-Einstein equation. So it showed that the diffusion coefficient is directly proportional to the absolute temperature and Boltzmann constant, but inversely proportional to the radii of the particles and the viscosity of the medium. Stokes-Einstein relation. The mobility of a molecule in cellular environment is affected by the following parameters, like the size of the molecule. Like an 8-fold increase of the size of a soluble spherical protein decreases diffusion coefficient by a factor of 2. Also depends on the viscosity, like membranes have much higher viscosity than cytoplasm. Then protein-protein interactions and binding to micromolecules can also slow down the diffusion. And if flow or active transport is involved in the movement of the probed molecule, the measured movement rate can become significantly higher than the theoretical diffusion rate. If we talk about electrochemical gradient, it's a gradient of electrochemical potential, usually for an ion that can move across the membrane. So the gradient consists of two parts, the chemical gradient or the difference in solute concentration across the membrane, and the electrical gradient or difference in charge across the membrane. So when there are unequal concentrations of an ion across the permeable membrane, the ion will move across the membrane from the area of higher concentration to the area of lower concentration through simple diffusion. Ions also carry an electric charge that forms electric potential across the membrane. So if there is an unequal distribution of charges across the membrane, then the difference in electric potential generates a force that drives ion diffusion until the charges are balanced on both sides of the membrane. An electrochemical gradient has two components. First, the electric component caused by a charge difference across the lipid membrane. Second, a chemical component caused by differential concentration of ions across the membrane. So, the combination of these two factors determines the thermodynamically favorable direction for an ion's movement across the membrane. Now, the generation of a transmembrane electrical potential through ion movement across a cell membrane can drive biological processes like nerve conduction, muscle contraction, hormone secretion, and other sensory processes. Right? So, by convention, a typical animal cell has a transmembrane electric potential of minus 50 millivolt to minus 70 millivolt inside the cell relative to the outside. Now we know that electrochemical gradients also play a huge role in establishing proton cations in oxidative phosphorylation in mitochondria. So the final step of cellular respiration is the electron transport chain. So four complexes embedded in the inner membrane of the mitochondrion make up the electron transport chain. However, only complexes 1, 3 and 4 pump protons from the matrix to the intermembrane space. So light-dependent reactions of photosynthesis basically pump protons into the hydroxyl lumen of chloroplast to drive the synthesis of ATP by ATP synthase. So the proton gradient can be generated through either non-cyclic or cyclic photophosphorylation. So this schematic shows the kinetic properties of glucose transporters. Now, GLUT4 activity is regulated by insulin-dependent translocation. Intracellular pool of GLUT4 in membranous vesicles translocate to the cell membrane when insulin binds to its receptor. Now, the presence of more receptors increases the Vmax for glucose uptake. It does not affect the KM, like does not affect the binding of the enzyme to substrate. Makes sense, right? And so when insulin signal is withdrawn, GLUT4 proteins will return to the intracellular pool. So GLUT4 is present in muscle and adipose tissue. So what is the fate of glucose in liver? Glucokinase will convert it to glucose 6-phosphate, then either glycogen synthesis or glycolysis or pentose phosphate pathway. We have talked about glucokinase and exokinase in the previous question. So if you see glucokinase as a Km very high, so it is not inhibited by glucose 6-phosphate. It is present in liver and pancreas beta cells. Whereas in case of hexokinase, the Km is pretty low. Okay, so it's inhibited by glucose 6-phosphate and is present in most cells. So exokinase has low Km and therefore can efficiently use low levels of glucose but is quickly saturated. Okay, as glucokinase is found in liver and beta cells of pancreas, glucokinase allows liver to respond to blood glucose levels. It has a high Km, so it does not become saturated till very high levels of glucose are reached. And at low levels, very little taken up by liver, so is spared for other tissues. So it is not inhibited by glucose 6-phosphate, allowing accumulation in liver for storage as glycogen. Now, glucose will enter the beta cell as blood glucose concentration will rise, right? And glycolysis will generate ATP, it closes to A plus channels in the cell membrane. It will stop outward transport and open calcium channels. An inward flux of calcium will cause exocytosis of insulin containing secretory vesicles. So, glucose will also stimulate new synthesis of new insulin. Fate of glucose in muscle, it will get converted to glucose is taken in glycogen synthesis or glycolysis. Fate of glucose in adipocytes, it's also self-explanatory. Glucose is taken in glucose 6-phosphate, then either to glucose 3-phosphate triglycerides, and on the other hand, insulin is also taken in by LPL and then fatty acids. So how is this metabolism regulated? So you can follow either two processes, catabolic or anabolic, right? So catabolic basically means breaking down molecules to generate energy, whereas anabolic means requiring energy for synthesis of molecules. The two pathways are kept distinct by regulatory mechanisms and sequestration in different cell compartments. Now, the pathways that contain recurring enzymatic mechanisms like oxidation reduction reactions, isomerization reaction, group transfer reactions, hydrolytic reactions, addition of removal of functional groups, and all of that. Okay. So, we'll not go in. Yeah. So, glycolysis, if you see, this is overview of the glucose metabolic pathways from glucose 6-phosphate to pyruvate, from oxaloacetic to glucose 6-phosphate, from glucose 6-phosphate to glycogen, from glycogen to glucose 6-phosphate, like TC cycle. So all these pathways must be very carefully regulated to keep the pathways going in opposite direction or proceeding simultaneously. So everything needs to be properly regulated. If there is any mishap happening at any point, it can lead to the breakdown of the system. So we have talked about this, the regulation of glycolysis. Glycolytic flux is controlled by need for ATP and for intermediates formed by the pathway. Control occurs at sites of irreversible reactions. Phosphofructokinase is a major control point, first enzyme unique to glycolysis. Hexokinase or glucokinase, pyruvate kinase. Then phosphofructokinase responds to changes in energy state of the cell, like high ATP levels inhibit. H plus concentration, high lactate levels can inhibit. Availability of alternate fuels such as fatty acids, ketone bodies, then insulin glucagon ratio in blood, and all of that. So we'll talk about phosphofructokinase. So this phosphofructokinase high ATP, when there is high ATP, lower affinity for fructose 6-phosphate by binding to the regulatory site, that is quite distinct from the catalytic site, right? Then there is high H plus concentration that will reduce activity to prevent excessive lactic acid formation and drop in blood pH. Then in case of citrate, citrate will prevent glycolysis by accumulation of the citric acid cycle intermediate to signal ample via synthetic precursors and availability of fatty acids and ketone bodies for oxidation. Now, phosphofructokinase as a regulator of glycolysis. Okay, we have talked about all of this. Basically, it activates PFK. One is activated by fructose 2,6 bisphosphate. It activates PFK1 by increasing its affinity for fructose 6-phosphate and diminishing the inhibitory effect of ATP. Phosphofructokinase 2, PFK2, is also a phosphatase bifunctional enzyme. So 6-phosphofructo 2 kinase activity decreased by phosphorylation, right? And fructose 2,6 bisphosphatase activity increased by phosphorylation. What is the hormonal control of all of these? What is the hormonal control of all of this? So glucagon or epinephrine, glucagon basically in liver and epinephrine in muscles, they can increase the cyclic AMP, cyclic adenosine monophosphate levels. It can activate the CAMP dependent protein kinase, and in liver this will lead to decreased fructose 2,6 bisphosphate, uh, 2,6 phosphate bisphosphate, and inhibit glycolysis, right? So this effect is opposite in muscle because epinephrine stimulates glycolysis. So in case insulin decreases calcium, not calcium, cyclic AMP increases fructose 2,6 bisphosphate and stimulates glycolysis. So move ahead. Now, if you talk about gluconeogenesis, so what is this process? It's a basically mechanism to maintain adequate glucose levels in the tissues, especially in brain. So, brain uses 120 gram of the 160 gram of glucose needed daily. Erythrocytes also require glucose. It occurs exclusively in liver and kidney. Now, glucose is synthesized from non-carbohydrate precursors derived from muscle adipose tissue, pyruvate and lactate, and then amino acids. So gluconeogenesis takes energy and is regulated. So converts pyruvate to glucose. Gluconeogenesis is not simply the reverse of glycolysis. It utilizes unique enzymes like pyruvate carboxylase, PEPCK, like phosphoryl pyruvate carboxykinase, fructose 1,6 bisphosphatase, and glucose 6-phosphatase for irreversible reactions. For this, 6 ATP equivalents are consumed in synthesizing one glucose from pyruvate in this pathway. Now the irreversible steps to gluconeogenesis. The first step by a gluconeogenic specific enzyme occurs in the mitochondria, right? So pyruvate will get converted to oxaloacetate in the presence of pyruvate carboxylase. So once oxaloacetate is produced, it is reduced to malate, right? So that it can be transported to the cytosol. And in the cytosol, this oxaloacetate is subsequently decarboxylated or phosphorylated by this phosphoryl pyruvate carboxykinase, second enzyme that is unique to gluconeogenesis. And the resulting phosphoryl pyruvate is catabolized by glycolysis enzymes in reverse until the next irreversible step. So in case of irreversible steps of gluconeogenesis, the fructose 1,6 phosphate is phosphate to fructose 6-phosphate. So in liver, glucose 6-phosphate can be dephosphorylated to glucose. It is released and transported to other tissues. and this reaction occurs in the lumen of the endoplasmic reticulum. Okay, this requires basically 5 proteins: glucose 6 phosphate transporter, calcium binding stabilizing protein, glucose 6 phosphatase, glucose transporter, and inorganic phosphate transporter. So basically, gluconeogenesis and glycolysis are reciprocally regulated. Okay, so fructose 1,6 bisphosphatase is the main regulatory step in gluconeogenesis, and corresponding step in glycolysis for 6 is 6-phosphofructose 1 kinase, and these two enzymes are regulated in reciprocal manner by several metabolites. So these substances are transported for chemical reactions, electric potentials at the membrane, or elimination from the cells. We know about all of this. We know about the cell membrane, how it surrounds the entire cell and cell organelles, all of that. And we know that there are peripheral proteins on the side. We have talked about all of this. Transport also we have talked about. We will not go into details. We have talked about all of this, diffusion and all of this. Gating channel also we have talked. We talked about this also, Fick's law. So factors that can be affecting the rate of diffusion can be lipid solubility, molecular size and weight, temperature. These are all molecular components, or thickness of membrane and surface area, membrane related, and concentration gradient, pressure gradient, electrical gradient. All of these are gradients. I have talked about the Fick's law. We have talked about all of this, facilitated diffusion. We have talked about all of this. We have talked about osmosis, osmotic pressure. We have talked about the different solutions: isotonic, hypertonic, and hypotonic. We have talked about all of this: active transport, passive transport. Okay, coming back to the this, we have talked about this, but if you want to see the differences, both of these are passive transport. Simple diffusion is for small molecules, whereas facilitated diffusion is for large molecules. In simple diffusion, no carrier is required, but for facilitated diffusion, carrier is required. Rate of transport is directly proportional to the concentration gradient in simple diffusion, whereas in facilitated diffusion, it is proportional to the concentration gradient till Vmax. And then we have these examples. So coming back to a question answer for today, which of the following cell surface receptor is involved in the uptake of glucose? It is glutes, definitely. Okay, so I'll wait for 5 minutes over here. If you have any doubt regarding this, we can discuss, or we'll move ahead to the next question after 5 minutes. Okay, so we'll go to the next question for today. So fructose 1,6 bisphosphate is broken down into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by enolase, aldolase, pyruvate kinase or isomerase.
Aldolase.
Right answer is aldolase. So we are focused mostly on glucose transporters and glycolysis in this session. So a little bit again on summarizing part. So as we know, glycolysis set of reactions that converts glucose to pyruvate or lactate, and is the first metabolic pathway to be elucidated, and hence is considered as a paradigm of metabolic pathways. It's also called EMP or Embden-Meyerhof-Parnas pathways, right? And the complete set of reactions occurs in cytoplasm of virtually every animal cell. And the entire process occurs without molecular oxygen, and it consumes 2 ATP and generates 4 ATP. Thus, the process results in the generation of 2 net ATP. So, the process also generates 2 NADH. What happens to pyruvate depends upon the presence or the absence of mitochondria in the cell and upon the availability of oxygen in mitochondria containing cells. Okay, so in case of cells like RBCs, like erythrocytes, cornea lens, no mitochondria, so pyruvate will get converted to lactate. In cells with mitochondria, now it depends on O2. If there is lack of O2, relative lack of O2, then pyruvate will get converted to lactate. If there is enough O2, then aerobic conditions, pyruvate will get converted to CO2 and H2, right? Then for the glycolytic pathway to continue, NAD plus has to be regenerated. So in erythrocytes that is having no mitochondria, and in mitochondria possessing cells under anaerobic conditions, NAD plus is regenerated from NADH during the conversion of pyruvate to lactate. And in mitochondria possessing cells under aerobic conditions, NAD plus is regenerated by either malate, aspartate shuttle, or alpha glycerophosphate shuttle, which transfer the reducing equivalents from NADH into mitochondria for electron transport chain, thus regenerating NAD plus cytoplasm. Now, what are the reactions? So, glycolysis will consist of two phases. In the first phase, glucose will be broken down to two molecules of glyceraldehyde 3-phosphate in a series of 5 reactions. And second phase, another series of 5 reactions will convert these two molecules of glyceraldehyde 3-phosphate into two molecules of pyruvate. So, phase 1 consumes 2 ATP and phase 2 generates 4 ATP. And the net ATP generated in the process is thus 2. This is the entire glycolysis schematic. So we have talked about the individual reactions. We will not talk about the reactions in such. We will talk about the facts about the reactions. This is the first intercellular reaction of glycolysis. Okay, so all these reactions happen in cytoplasm. It requires ATP, that is magnesium, also cofactor, and this is one of the investment reactions. And the phosphorylation of glucose traps the glucose inside the cell, and the reaction is considered irreversible. So hexokinase is the KM of glucose for glucose of less than 0.1 molar, which means it has a high affinity. It is also inhibited by the product glucose 6-phosphate. Whereas liver hepatocytes and pancreatic beta cells contain another enzyme, glucokinase. Glucokinase is a KM for glucose of about 10 millimolar low affinity, and glucokinase is not inhibited by its product glucose 6-phosphate. It's induced by insulin, and the levels of glucokinase in the liver of untreated type 1 diabetes are lower than normal. So hexokinase and glucokinase are isoenzymes. So therefore, irrespective of the isoenzyme catalyzing the reaction, the K equivalent of delta G for the reaction will remain the same. The next reaction will phosphoglucose isomerase. In this case, the reaction will be carried is reversible and functions in both glycolysis and gluconeogenesis. So it means the conversion of aldose to a ketose. Next case, a reaction is rate limiting step of glycolysis. It is irreversible and the completed step. It is an allostatic enzyme and also a major regulatory enzyme. So we have invested a second ATP molecule over here in this case. Reaction 4 will be aldolase, but the question was all about fructose 1,6 bisphosphate will get converted to dihydroxyacetone phosphate DHAP and glyceraldehyde 3-phosphate G32. So we now have two phosphorylated trioses, right? And only glyceraldehyde 3-phosphate is used in glycolysis. So therefore, dihydroxyacetone phosphate needs to be converted to glyceraldehyde 3-phosphate, and this occurs in the next step. So triphosphate isomerase basically catalyzes the interconversion of DHAP and glyceraldehyde 3-phosphate, and because of the interconversion, one glucose molecule can be converted to two glyceraldehyde 3-phosphate molecules. Then the glyceraldehyde 3-phosphate dehydrogenase will convert glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate, and the enzyme oxidizes the number 1 carbon aldehyde and then adds a phosphate group. So we have an acid anhydride in the product 1,3-bisphosphoglycerate, and the phosphate on number 3 carbon is not a high energy bond. So we have used an NAD plus for the oxidation reaction, and cell has limited amounts of NAD plus. So somewhere along the line we have to regenerate it or glycolysis will stop. So this reaction is a target for arsenate basically. So arsenate will resemble inorganic phosphate. In the presence of arsenate, the product of reaction is 1-arseno 3-phosphoglycerate, and this product is unstable and decomposes into arsenate and 3-phosphoglycerate and no ATP formation. So after this step, glycolysis continues. So the enzyme contains an essential thiol group at the active site. Iodoacetic acid is also an inhibitor of this reaction, and it reacts with the active site SH group and inhibits the enzyme. So phosphoglycerate kinase is the first step of energy production. This is referred to as a substrate level phosphorylation as opposed to oxidative phosphorylation that occurs in mitochondrial ATP production. And we have recovered both ATP that were invested. So each glucose gives to two phosphoglycerate molecules. Then we have phosphoglycerate mutase and then enolase. Enolase will catalyze the dehydration of 2-phosphoglycerate to form phosphenol pyruvate. Okay, and this reaction will be inhibited by fluoride. And we have pyruvate kinase at the end. It will catalyze the transfer of the phosphate from PEP or phosphenol pyruvate to ADP to generate ATP MPI and pyruvate, sorry. And then the reaction is called substrate level phosphorylation. We have discussed why is it called substrate level phosphorylation as well. So this reaction completes a part of glycolysis that is common to both anaerobic and aerobic metabolism. So under aerobic conditions in the presence of mitochondria, pyruvate can enter the citric acid cycle. NAD plus is generated by malate aspartate shuttle or by alpha glycerophosphate shuttle. And under anaerobic conditions, in the absence of mitochondria, pyruvate is reduced to lactate, which basically is lactic acid fermentation, and NAD plus is regenerated. Now we have anaerobic glycolysis. So when pyruvate cannot be oxidized within mitochondria for some reason, pyruvate is reduced to lactate by lactate dehydrogenase. So you have talked about all these steps. If we can talk about the clinical considerations now. So when there is a high blood level of lactate, okay, blood pH can be less than 7.5 in severe cases. So what happens in case of lactic acidosis? So most tissues can convert lactate to CO2 and H2 through the TCA cycle, but if the oxygen level is very inadequate, then cells must rely on glycolysis for ATP production, and a decrease in ATP enhances glycolysis at the level of PFK1 and produces more pyruvate and hence lactate, right? What happens? Accumulation of plasma lactate may be secondary to tissue hypoxia, severe anemia, mitochondrial enzyme defects, and inhibitors of oxygen transport. Again, bicarbonate is usually administered to control acidosis associated with lactate overproduction. Dichloroacetate can be used as a drug to treat lactic acidosis because it activates pyruvate dehydrogenase in mitochondria, which facilitates the conversion of pyruvate to acetyl-CoA so that lactate production is decreased. Then strenuous exercise will lead to anaerobic glycolysis, and this causes overproduction of lactate. In case of diabetes, insulin activity is low, Consequently, glucagon levels increase. So, thus glucagon insulin ratio is higher in diabetes. So, there is no change in glucose uptake in liver because this tissue does not express GLUT4. The insulin responsive facilitated glucose transporter. So, in skeletal muscle and adipocytes, glucose uptake decreases because of the absence of insulin-dependent recruitment of GLUT4 into the plasma membrane. So, in liver, glucose is not metabolized effectively because of this, and this also results in an increase in gluconeogenesis, producing more glucose from gluconeogenic precursors alanine and glycerol. So these precursors come from muscle and adipocytes as a result of increased protein and amino acid breakdown and lipolysis to provide energy because of the decreased utility of glucose as an energy source. Then if we talk about glucokinase deficiency, mutations in the glucokinase gene leading to inactivation of the enzyme can be associated with a form of non-insulin dependent diabetes mellitus, uh, called the maturity onset diabetes of the young, and complete absence of the enzyme activity will also lead to type 1 diabetes because of the lack of insulin secretion in response to the blood glucose, and this condition is associated with severe hyperglycemia, and mutations in the gene leading to increased activity of the enzyme will cause hyperinsulinemic hypoglycemia. Okay, now then we talk about pyruvate kinase deficiency in hemolytic anemia. So any kind of genetic defect causing a 5 to 20% reduction in the red cell pyruvate kinase levels. It's a rare but most common genetic disorder associated with glycolytic pathway. So there can be other glycolytic enzyme defects as well. Like there can be deficiencies in the enzyme activities of phosphofructokinase, phosphoglycerate kinase, phosphoglycerate mutase, and lactate dehydrogenase to represent genetic defects in glycolysis. All of these have certain common clinical features: exercise intolerance, myoglobinuria, hemolytic anemia, and all of that. Okay, so coming back to the question that we started with, uh, fructose 1,6 bisphosphate is broken down into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by, so the answer is aldolase. Okay, so this was the last question for today. If you have any doubts regarding this, we can discuss it for some time. Otherwise, we can conclude the session for today. Any doubts? I'll stop sharing this screen. Okay, so in case if there is no doubt, we can stop the session for today, and we'll meet again next Tuesday, same time. Thank you.
Thank you, ma'am.
Thank you, ma'am.